Hello there, and welcome to another episode of Innovation on the Loose, streaming from the DeBakey CV Education studios at Houston Methodist Hospital. And today I'm delighted to have in the studio my special guest, Dr. Darren Kraemer. He is founder and CEO of Light Matter Interactions, a Canadian-based company pioneering laser surgery technologies based on novel picosecond infrared lasers invented by researchers at the University of Toronto. Dr. Kraemer, welcome to the show.
Thanks, Stuart.
So I know yourself and myself have known each other for about 6-7 years, so I'm somewhat familiar with the technology. But you got to tell our audience, what is a picosecond infrared laser and what applications are they finding in surgery? And I believe you have a few slides as well to help with that, right?
I do. Thank you for the kind introduction. I'm going to attempt to briefly go through the history of this technology, which we began to develop about 15 years ago while I was a PhD student at the University of Toronto in the group of Professor Dwayne Miller. We were working on some molecular dynamic spectroscopy, trying to understand how the energy transfer timescales of vibrational modes in liquid water. And in the process of doing this, we were also developing somewhat exotic lasers to do our experiments. And Professor Miller had this great idea for a new kind of surgical laser in which we would target these vibrational modes in order to produce a tissue disruption mechanism that would not burn tissue, it would not melt tissue, it wouldn't ionize. And you know, it's been quite a journey. So the problem that was first presented is the issue of damage to the tissues caused by surgical devices. So you think the scalpel just divides tissue. It's not so simple. It stretches and shears the tissue and tears it on a microscopic level, and this leads to damage. It doesn't coagulate. If you introduce a form of energy, either electricity or laser energy, typically this results in burning and damage to the adjacent tissue, which all of these effects lead to scarring, adhesions, and delays in healing. So this is a video of an erbium YAG laser being used on skin of a mouse, and you can see that the tissue is shrinking because of the heat, it burns, and this is not a suitable device in order, if you want to perform a minimally invasive incision. So what we came up with, we call it the PEARL, or the picosecond mid-infrared laser. And this is what we think might be the ideal surgical laser technology for precise tissue removal, for minimally invasive procedures where you don't want to damage adjacent tissue, and for frictionless incisions. So the key attributes that are required for this is a wavelength that absorbs very strongly in tissue. We are targeting the OH stretching modes of water, which is a major component of tissue. And then at the same time, there are lasers that do this already. What makes ours different is we have a very short pulse duration. It's faster than the time it takes for heat to diffuse out of the irradiated zone, and it's faster even than the time it takes for acoustic shock waves to propagate. As a result, our laser goes in and it energizes the tissue, and it uses the water to very quickly shoot the tissue out like a propellant. And because of the long wavelength, it's non-ionizing. Because of the time scale of this process, it's effectively cold ablation or not thermal. And it really localizes all the energy so that we can achieve tissue disruption and vaporization. Here is a video in which we see a comparison. On the bottom, this is a vocal cord. And there's a CO2 laser cut, which is the conventional technology that's used now. Above it, you can see our PEARL in action. And the difference is quite obvious in terms of the amount of burning and charring to the tissue. So I'm going to show you a number of examples here. This is on-contact cutting with a fiber optic. We have to use sapphire because of this wavelength. Most glasses don't transmit. And you can see wherever we touch the tissue, it basically disappears. And so even though this was handheld, the resolution of this kind of surgery is quite extreme. Compared to other laser technologies, if you imagine a long pulse laser is kind of like using a magnifying glass with the sun. You focus the light down to a very small spot, but at that spot it's absorbed, the temperature rises, eventually you get melting or burning. If you pack that energy into a short enough pulse, you can create a large shock wave that also damages adjacent tissue. Around the time I was doing my PhD, there was a lot of interest in ultrafast lasers, which are femtosecond or picosecond lasers. And here you pack all that energy into a small space and time, and the electric field is so strong that when you expose the tissue to it, it rips the electrons out of the tissue and it ionizes and fragments into tiny particles. This is really useful if you want to cut brittle materials like semiconductors and Gorilla Glass, but it's not really suited for biology just because of the high energy electrons, which make this somewhat mutagenic. Of course, it is used in eye surgery and in some other procedures. What we came up with is quite different, and we've been working on commercializing this now. In the process, we've jumped through many hoops. Here you see some examples of the different in the remaining tissue after cutting with a conventional laser that heats or an ultrafast laser or our laser. And what you see is we've stained the tissue to show live cells versus dead cells. The thermal laser causes a large zone of collateral damage, whereas our laser, really, you only see a few dead cells. Maybe they've been cut in half. Femtosecond laser nukes everything, and it might be hard to see, but there's a sort of continuous line of dead cells, and this limits the healing. If you zoom in, the bottom row is electron microscope images. This was a pig jaw that we machined, and the difference is quite obvious. With our laser, there's still intact collagen waving around on the surface, and if you zoom into that, you see that it hasn't been denatured. So you can imagine that there are numerous areas in which this sort of technology might be useful, and I've listed a few of them here. But essentially, wherever there's a need for more precise energy to disrupt tissue, when you need high resolution, when you need to avoid damaging delicate structures nearby, this is where our technology may have some advantage. So one of the applications we've investigated is ophthalmology, and here we are attempting to compete with a device that has a very sharp ultrasonic vibrating needle, and if we can make something that is as effective but has no moving parts, then of course this brings this technology into the modern age. Other applications of this are in cosmetic surgery and dermatology, particularly skin incisions is something that we've investigated. We did some early studies with different research groups. Here you see a comparison of wounds that are made on mouse skin using our laser, using a scalpel, using electrosurgery. Actually, it was rat skin. And if you look carefully at the bottom, you'll see that with our laser, there's practically no scar. There are 2 little marks from the sutures. But by comparison to the scalpel, it seems to heal very well. And the reason is likely that we're able to make a perfectly straight incision so that when we bring this tissue together, there's no gaps for the scars to fill. We did a healing study comparing our laser to a conventional laser, expecting to see improved healing due to the lack of thermal damage, but we also included a punch, which is a mechanical blade, as a comparison, and we were very surprised to find that our laser actually heals faster. And when this group looked at the protein signals of scar formation, they found it was much lower with our laser. So this is very encouraging, and it is one of our goals to master skin incisions so that we can provide scar-free surgery to patients and cosmetic surgeons. However, this is, it's not as easy as that. It seems there's a lot more that goes into wound healing than just making the incision. But this is an area that we're still very interested in pursuing. We also think that there are applications in microliposuction for this laser as well, as it is very precise. And then you can imagine that there are lots of uses. The last time I came to visit you, we talked about the scar from thyroid removal surgery. This is just the tip of the iceberg. So another application that we've been looking at, and is the reason for my visit here in Houston, is vascular surgery. Particularly, we think that there may be some benefit to our laser for treatment of peripheral artery disease, particularly crossing chronic total occlusions. And peripheral artery disease, of course, affects millions of people. And it can be very challenging, and something like 20 to 30% of procedures to unclog those arteries in the legs fail because the current devices are very flexible and they cannot push through hard occlusions. So we've been thinking about this. Others have used lasers in atherectomy. They typically use ultraviolet lasers, excimer lasers. And if you can imagine shining a very, very bright ultraviolet light inside of your arteries, there is going to be a sunburn. And we suspect this may be one of the causes of restenosis. So the question that we have to answer now is, can our laser improve the safety and the efficacy of this procedure? This is an interesting paper that we found where we show that the cavitation bubbles caused by these large energy ultraviolet pulses can actually create mechanical damage inside of the vessels, whereas our approach is going to be significantly more gentle. Partly this has been inspired by an old experiment that we did a long time ago. When we first were able to couple our light into fiber optics, we wanted to see whether or not the laser was actually doing any of the work of drilling. So we took a piece of fat. Here we're holding it on a toothpick. And with the laser off, we try and poke our little sapphire fiber in, and it just moves out of the way. Now we turn the laser on, and our laser goes through with practically no friction whatsoever. So we've known about this phenomenon for a long time. I'll skip that. And more recently, we've been able to apply this to arterial plaque tissue. In this case, you can see that we are attempting to cross this very, very hard occlusion by hand. And if you tried to do that with a guide wire, of course, it would just buckle. We then took this to one of the hospitals in collaboration with the group there. Built a little benchtop model so that we can measure the force that's involved in this crossing. And here you see two interesting graphs. On the left, as we push into the tissue without the laser on, the force increases. And if we turn the laser on, of course, there's significantly less force, orders of magnitude less. On the right, we do it a different way. We advance until the force reaches a certain threshold, and then we turn on the laser, and you can see this very quick drop in the force. So we have this technology that can do frictionless piercing in tissue. This is a cross-section of some plaque that we machined, and you can see the area that we removed is abnormally symmetrical. So we thought that was a very cool result. Ultimately, our intention is to provide the surgeon with a device that gives them the ability to visualize what's going on inside of the artery, gives them the ability to guide the fiber to specific points, and then either pierce or debulk. And we've built various prototypes of this so that we can continuously validate this. Here you can see a wet lab experiment where we have some tissue in a test tube, and we're taking our little tube robot fiber optic, and wherever we touch the tissue, it turns to dust. In this case, right now we're not aspirating, so it kind of gets cloudy. Once we turn on the aspiration, these tiny little particles effectively get removed. They disappear. That'll happen any second now, or maybe I'll just wind forwards. There you go. And on the right, you see the image from the endoscopic camera that we've incorporated into this. So there's still a lot of work to do here. We've also been thinking about how a similar device might be used to treat thrombosis. And on the right, you see a little steerable catheter that we developed that made that nice little hole in our thrombosis model. So that's the vascular surgery application. Of course, you can continue to imagine all the different places where a surgeon might find this technology useful. One of them is neurosurgery. I think this next video might be a little gross. This is a lamb brain we got at the butcher shop. And I was talking to neurosurgeons, and they introduced me to keyhole surgery. So I decided to make the world's smallest keyhole as a joke. You'll see that this little blue object is a 250 micron diameter sapphire fiber. And if you just poke the surface, it doesn't do any damage. But if you turn on the laser, anything you touch turns to a kind of liquid dust. And the other device is just a little needle for aspiration. I'm going to fast forwards a little bit. But you can see we can make these perfect cavities, and they can be smaller, wherever we touch. We could go deep into the brain, making almost, no collateral damage compared to some other devices. And at the end of this, I cut a little keyhole that's only a few millimeters deep. Most brain surgeons don't really appreciate this joke. So we imagine that this might be useful. Currently, lasers are used inside the brain. An example is Visualaise, where they use MRI guidance to put a fiber optic inside the brain, and then they use the laser to heat the tissue, and they watch on the MRI as the heat spreads, and once it gets outside of the sort of region of interest, they turn it off, they wait, they turn it back on. They want to reach a certain temperature for cell apoptosis. I think that we can build a device like this that has significantly higher resolution. They're operating on a centimeter scale. We'll be able to operate on a millimeter or maybe even sub-millimeter scale eventually. We can make these fiber optics as smaller than 100 microns, the size of a human hair. And you can imagine them going in frictionlessly anywhere inside of the body to do their work. The difference being, though, that instead of using heat, we'll actually have to go and touch all these various regions with our fiber. So that makes it a little bit more complicated. In general, there have been huge advancements in medical imaging. And as the resolution of medical imaging increases, it seems that the devices needed in order to manipulate the tissue require a matching increase in resolution. And this is where I think our technology has a lot of value. Not just to see what's going on, but also to treat it at that scale. I like to show this video that we made a long time ago. On the left is a Da Vinci robot attempting to peel a grape.
Actually, one of my favorite videos that I've seen. Phenomenal.
They put this on YouTube, but you can see the first thing that happens is they damage the grape skin. On the right, you see our laser being delivered by a scanning system. We're able to cut halfway through the grape skin. We're able to cut all the way through, and even the clumsy hands of an engineer can pull out an absolutely perfectly circular bit. And then, of course, we can peel the entire thing like one might peel a potato. The possibilities are endless. So I showed this to an investor a long time ago, and they put their arm around me and said, I don't think there's a big market in peeling grapes. It's up to us to demonstrate the usefulness of this technology so that we can put it in the hands of surgeons. This is, I think, a technology push that we've been doing for 15 years, but somebody has to do it. There's all this potential. We really believe that we can make useful devices out of this. We've shown that we can do that in several fields, and the goal is to just continue to take devices like this to market. More recently, we've been thinking that, could we figure out a way to go anywhere inside the body without creating a significant wound or significant damage and deliver small amounts of locally activated drugs to improve treatment for cancer and other conditions. If you follow some of the research, people use very, very small tapered capillaries to make injections inside of mouse brains and things like this. But for humans, we're limited to standard gauge needles, something like a 30 gauge needle, just because the scale is larger. And so what we've tried, just skip that. What we're working on is a laser-assisted injection device that allows us to go to a targeted region of tissue using our mechanism, and then while we're there, we can create a little void place some drugs which won't be shot out because of the high interstitial pressure. We can also photo fix these drugs. We can do photodynamic therapy because we will have an optic already in there. And we can even do photothermal disruption. There's a lot of possibilities. I'll skip this one. At the moment, our prototype device looks something like this. It's about 250 microns in outer diameter, so a little smaller than a 30 gauge needle. And you can see that we're able to squeeze the liquid right out at the tip. These are the options when it comes to biopsy needles. I'm sure there are many, many more. We have been thinking for a long time how we might apply the same technology to do the complete opposite. Instead of delivering a drug, if we can go in and we can liquefy some of the tissue, we can extract it without cooking it, without fragmenting the particles, we can likely perform a very, very fine needle biopsy. And then another topic that we've been investigating for a long time and that we have spun off into a separate company is biodiagnostic applications, in which we want to do almost real-time molecular testing on tissue by taking advantage of our ability to put solid tissue into the gas phase and then analyze it with a mass spectrometer. There are many situations in which a clinician wants to identify the tissue, but they don't have time for a frozen section, which takes about 20 minutes, or much slower molecular tests. And this is a critical component of personalized medicine. If you want to stratify your patients, you need to be able to do molecular testing, and you need to do it fast, and you need to do it cheap. And of course, there are many very sophisticated forms of molecular tests that are available at the moment. So the x-axis is time, and at 20 minutes is sort of the cutoff for useful point of care. There seems to be a technology gap here for rapid and accurate molecular testing, and we've been developing a product that we think fills that need. I don't have a lot of time, so I'm just going to show you this little video to explain how it works. You imagine the surgeon has this device in their hands. They vaporize a tiny little bit of tissue, which goes into the gas phase without destroying the intact biomolecules. We then aspirate this tissue through a mass spectrometer, which sorts and counts the molecules to produce a spectrum that's unique to the specimen, like a fingerprint. And then we use machine learning to compare this fingerprint to a database of fingerprints from known tissue types. And within about 10 seconds, we're able to identify the tissue. So the whole setup sort of looks something like this. As an example of the accuracy of this, on the left you see 3 types of pediatric brain cancer, which our device can distinguish almost perfectly with only 10-second sampling time. And then medulloblastoma has 4 known molecularly distinct subtypes that are very difficult to distinguish under frozen section because morphologically they look identical, and we're able to classify these with 97% accuracy. And of course that's a video, and it just crashed.
Well, I think it's actually a really nice video to end on and segue into Q&A. I mean, you're vaporizing the tumor there and you're analysing it. It's phenomenal. So I've got a bunch of questions. I'm going to go back to the start. So why was this study done in the first place? You said you were looking at vibrational modes in water, right? Doing some water spectroscopy. Why were you even looking there in the first place?
So I was trying to get my PhD.
Okay.
And my background was engineering. Mm-hmm. And in my 4th year, I met Professor Dwayne Miller, and he taught a course called Atom Solids and Molecules, and it was riveting. I was fascinated, and he kind of sucked me into grad school. I had no intention of doing it. And when the time came, I wanted to do physics because I had done engineering physics. And he said, no, the physics is full this year. You have to do chemistry. And I said, okay, is that going to be hard? And he said, no problem. And then he had been working on this problem, trying to understand water being the most ubiquitous solvent in biology, and you think everyone understands water, but that's not the case. On a microscopic level, on a quantum mechanical level, there's a lot of unknowns. It has these miraculous properties. It's very unique. It exists, on Earth in all these phases of matter. So it's a fascinating subject. And we were trying to do time-resolved vibrational spectroscopy on water molecules to ping the vibration of the OH stretch and see how that mode then transfers into other modes. And some people believe that water has a memory. Ultimately, the purpose of the experiment was to measure the structural memory of water. And it turns out that it is very short. Something like shorter than 100 femtoseconds, which is different than a lot of other liquids. And so water has these unique properties, but this is where the great insight happened. If we can put the energy into the OH stretch, it very quickly becomes low frequency modes, which ultimately are motion.
Got it. So the energy you're putting in, it's not like breaking bonds, it's vibrating that OH bond.
That's right.
Okay, to such an extent that, like, so where's that? So you've got all these vibrations. So how does that then translate to everything kind of ripping apart?
Well, if you can imagine, the water molecules are not isolated. They have hydrogen bonds, up to 4 hydrogen bonds. And all the other molecules in tissue are sort of interconnected by these water molecules. So we put the energy into the OH stretch. It absorbs very, very strongly. And now that energy quickly and efficiently becomes these sort of long-range motions in between molecules. And eventually, you get a super rapid heating. And that just kind of rearranges all the molecules on a very, very fast time scale. All those bonds are broken, and it contains that energy, but there is, of course, a reaction to it. So you rearrange everything, and in the case of an air interface, that tissue that remains just gets shot out by this water.
And just to remind the audience again, what is the wavelength and the pulse duration?
So the wavelength can be anything that targets one of these vibrational peaks in water. The highest absorption occurs around 2.95 microns. And if we want to be faster than the timescale for both thermal diffusion and acoustic diffusion, the pulse duration needs to be on the order of a few nanoseconds or hundreds of picoseconds.
Got it. Now going into the system itself, again, I know you have different systems for different applications, but it seems a lot of this could be based on the flexibility of the fiber optic probe. So maybe talk to us about that. Like, what are the technology limitations there? Is there anything that you're trying to overcome with research and development?
So fiber optic coupling at this wavelength is a known problem, only because most fiber optics are made out of glass and silica-based glass, and it doesn't transmit at this wavelength. It absorbs very strongly, and you cannot use it. It will damage. So this is one of the limitations. There are glass technologies that do transmit at this wavelength. We discovered early on that sapphire fibers were transmitted very well and were also mechanically resilient enough that they could withstand repeated use. So you saw some of the fiber optics in the videos. We clean them by dipping them in water and turning on the laser. Because sapphire is so mechanically hard, it's biocompatible. It's really an ideal material. However, growing fiber optics out of sapphire is complicated, unlike glass, where you can just pull kilometers or miles, sorry, we're in the US, of fiber. For sapphire, you typically need to grow it as a single crystal. And this leads to a high cost and limitation. So we've spent a lot of time looking at other potential fiber optic technologies, zirconium fluoride glasses, germanium oxide glasses, and each of them sort of have their pros and cons. So a lot of what we do is we try and conceive of the surgical instrument and then put it together based on the various technologies that we have in our toolbox. Capping other fiber optics with sapphire, things like this, trying to make an assembly, but in order to achieve this very high resolution surgery, we're space limited. Ideally, it is just a single, hair-sized device that the surgeon is holding in some sort of injection molded plastic handle.
Got it. And maybe you can comment on lasers that are currently on the market for medical applications, and as a follow-up to that question, what applications are there out there which current lasers just cannot go into because of limitations and where the power laser could go into?
Ultimately, lasers are competing with lots of other technologies. The advantage of a laser is that you can focus it very tightly. You can deliver it at a distance using fiber optics or using projection. You can scan it very quickly. However, scalpels and drills and oscillating saws are relatively inexpensive by comparison. So it's always a question of where's the value proposition? Is it faster? Is it more effective? Is it safer? And when we can collect, enough value for a particular procedure, then we can move forwards. But because of limited resources, this technology may be considered a platform, but we can only investigate so many applications, so we have to stick to the bigger ones. So in terms of other lasers, that's another hour of discussion. Thermal lasers are used in all sorts of things, from treating the surface of the skin to cauterizing and coagulating. You have laser lithotripsy to treat kidney stones, which is, again, each type of laser that's been developed seems to have found a niche in medicine. Whether they're continuous wave diode lasers used by dentists on the gums, or ultra-fast lasers used by ophthalmologists to create these tiny little perforations inside the eye so they can just go in and sort of make those remove structures with perfect symmetry and things like this. It's a fascinating field, and the hope for our technology is that we will find our place in medicine.
So I'm just going to move on to like tissue types. So it sounds like more tissues that have more water, it would be absorbing this and interact with it more. Is that correct? And just talk me through the different types of tissues that this can work with.
I guess the ideal tissue is very soft. It has lots of water in it. There's not a lot of vasculature, because when we do cut blood vessels, they don't cauterize or coagulate, they bleed. Mm-hmm. So ideally, inert and soft tissue will be disrupted most easily. As the tissue gets harder and likely contains less water, we have to use more energy in order to exceed this threshold where the effect occurs. So we've shown that we can very accurately cut bone and tooth, but our laser technology is new. We are power limited. Mm-hmm. And so the speed isn't yet comparable with mechanical tools. The dream is that one day we'll be able to exceed the speed of a dentist drill. You imagine the dentist drill removes a lot of tooth, but you don't use it continuously. The dentist goes bzz, 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 or it'll burn the tooth. We could potentially use our laser continuously and scan it, project it under machine control and sort of make the perfect structure to help reconstruct bones. You can imagine, high resolution carpentry now available, but we have a lot of work to do in order to develop the necessary power to make that practical.
So say you had like an open skull kind of procedure where you want to get through the skull. Would your system be able to do that, even though there might be a time delay on it?
I think a human skull is quite thick. Mm-hmm. Whether or not you need all of these advantages in terms of the healing on the skull, I think is questionable. Mm. So you could imagine if we need to get inside the brain, we might just drill a little hole in the skull, leaving a margin of error, mm-hmm. and then use our laser to remove the final little thickness, possibly with OCT or another imaging system, so that we can, very finely determine when we're getting to the next layer, and really just wield this device where it's most needed.
Now, with this system, I think what really piqued my interest, a few years ago was the thought of scarless surgery. I mean, wow. Can you maybe just comment a little bit on your progress in that area? And I know we talked about thyroidectomies and potentially reconstructive breast surgery, but what have you been looking at?
We've been looking at ways in which to reliably make incisions in thick skin. Skin near the surface tends to be a little dry. But what we've realized and what we've found is that there's a lot more to, like I said before, there's a lot more to wound healing than just making the incision. You can make the perfect incision, but if you don't close it up properly, then it's going to form a horrible scar. So aside from applications like keloid scar formation, we have a lot of work to do to validate this claim that we can actually perform a practical scar-free incision, on a large area of human skin in a person that heals while they're also living their life, rather than floating in some sort of weightless chamber and not moving for 6 months. I think that it's likely possible that we will crack this problem in the future if we put enough resources into solving it. But we're forced to address the lowest hanging fruit procedures first. And so something like vascular surgery, I think we have, it'll be much easier for us to make a useful device for surgeons there at the moment. Plastic surgeons are masters at hiding scars. And ultimately scars are, at least outside the body, are mostly cosmetic. And obviously you want to avoid adhesions inside the body. Yeah. But in order to, in the early stages of technology commercialization, we really get very few chances at bat, and we have to hit home runs. And so in order to realize scar-free surgery, we have a lot of work to do. We need to increase the power of our lasers so that we can make these incisions faster, because the plastic surgeons can't stand around waiting for the laser to do the work that they can do within seconds using a scalpel.
It's understandable. And I guess the final question is more of a comment, or I'd like you to comment to the audience is, so you're down here from Toronto, you're doing basically a preclinical study at MIRI at the Bookout Centre, which is here at Houston Methodist. And we have Dr. Tricia Roy, who's a MD PhD surgeon here in the department. Any comments? Like, what was your experience been like working at MIRI at the Bookout Centre in that interaction?
It's been absolutely fantastic. Dr. Roy is very enthusiastic and seems very brilliant. There's no substitute for expertise. As engineers and scientists, it's our job to try and understand our customer. And the only way to do that is really to interact with them and to be exposed to this. So this was a wonderful opportunity to test the device that we've been working on for the last 2 years in a realistic use case scenario. And the setup here was absolutely perfect. And I don't think we could have done an experiment like this as easily anywhere else.
Last one. Well, appreciate you saying that, Darren. I think when we first met, I think my goal was to get a pearl laser system down here at the Med Centre as soon as possible. And obviously the pandemic hit, but I think we hit that milestone yesterday. And we officially now have, we had the pearl laser system here at the Med Centre, first time ever. So breaking ground there. Well, thank you very much for coming all the way down. Thanks for being in the studio. And I'd like to thank the audience again. If you have any questions, feel free to put them in the comments below. And you can always reach out to either one of us with any questions you may have. Thank you again. Goodbye.